the kind words, Sach, and good afternoon, everyone. Well, firstly, I'd like to thank Sach and Sylvia for inviting me to present my work today. It's really a pleasure to be speaking in this seminar series. So today I will talk about learning from data in single-cell transcriptomics, or in short, single-cell RNA-seq. I've had a terrific time working on this project with my PhD students and postdocs, in particular Philippe Boileau, Nima Hijazi, Fanny Perraudot, David Erisso, Hector Roux de Bézieux, Kelly Street, and Koen van den Berg. And special thanks to my collaborator of about 20 years, John Nye, for motivating most of my work in this area in high throughput genomics. And I'd also like to acknowledge my collaborators in statistics and computer science, Livan Clément, Elizabeth Purdom, Jean-Philippe Vert, and Nir Yosef. My group and I were particularly excited to see that some of the work that I'll present today was actually used to produce the first comprehensive atlas of mammalian brain cells, and the whole package, the whole project is published in a special issue of Nature on the Brain Initiative Cell Census Network. So it doesn't have much of the methodology in these articles, but at least the applications to building this atlas of the brain. So here's an outline of my presentation. So it's essentially a survey of the main steps in a workflow that we've developed to analyze single-cell RNA-seq data with a view towards studying how stem cells differentiate. So I won't spend too much time on any of these topics, but I'll just give a sense of the methods we've developed in each case. But let's start with the biological question that motivated our work. So our collaborators in the Nye lab are interested in studying how stem cells differentiate in the mouse olfactory epithelium. And their work has very important potential application, in particular to the prevention and treatment of neural tissue damage and degeneration. So you can think Alzheimer's disease applications to diseases like Alzheimer's. So they're focusing on a particular type of stem cell, which is called horizontal basal cell, in short, HBC. And they use the p63 protein as a molecular switch. So under normal conditions, HBCs self-renew. But when the p63 gene is knocked down or targeted in some other way to remove its function, the HBCs start differentiating into a range of cell types that include neurons and subtentacular cells. So to study how these HBCs differentiate in the olfactory epithelium, the Nye lab performs a range of RNA sequencing experiments at single cell resolution in mice that were wild type, but also that had the p63 gene knocked out. So here's a picture of a mouse's head. You have the olfactory epithelium here in blue-gray. And let's zoom in on the olfactory epithelium. And here are the main cell types in this tissue. At the top here, this is the portion that corresponds to what's exposed to the exterior, the odorants. So we have neurons at the top, and at the base here, we have horizontal basal cells. So under normal conditions, the HBCs self-renew. And if the gene is knocked out, they start differentiating into globose basal cells, GBCs, and different types of neurons and subtentacular cells at the top. So here's a little cartoon of what single-cell RNA-seq does. I won't spend much time at all here because I would assume most of you are familiar with it, but just in a nutshell, you first isolate single cells, you extract their RNA, you sequence the RNA, and then you map back the small reads to the genome. And the main idea in these experiments is that the number of reads that map to a particular gene is a measure of the expression level of that gene. So after much pre-processing, which I won't discuss, such as image analysis, base calling, and sequence alignment, we have read counts. And our entry point will be a matrix like here, where rows correspond to genes and columns correspond to single cells. And the entries of the matrix are read counts. So we'll use data of this type to address a range of questions that are of interest to collaborators, including what groups of cells are present in these olfactory epithelium cells, what genes are differentially expressed between the different cell types, how do these cells, in particular the HBCs, differentiate from one group to another, and also what are the genes involved in the differentiation process. So on purpose, I'm starting the presentation with a fairly small data set, which comprises only, by today's standards, only 600 cells. Nowadays, our collaborators have data sets that are more in the tens of thousands of cells, but I thought it'd be easier to illustrate the main points using this smaller data set. And also these days, we're looking at other data modalities than single-cell RNA-seq. We're looking at ChIP-seq, for example, to study also the role of chromatin modifications in the differentiation of these stem cells. So these bar plots here summarize the experimental design that we have. Each bar plot corresponds to a batch. So a batch represents a technical grouping of the cells. So by sequencing run or day of experiment, it could be a variety of different definitions of batch depending on the experiment. But batch reflect technical variation. So each bar is a batch, and the height of the bar reflects the number of cells in the batch. And then we've colored the bars by biology, and biology here is whether the cells are wild type or knockout. And the Nye lab also used tamoxifen treatment to prove the resolution of the differentiation process. So we also have the number of hours after tamoxifen treatment. So you can see right away here that there's a design flaw that you have batch effects that are nested within biological effects. So we'll have to be mindful of that at the normalization stage, because if we just try to remove batch effects completely, we may remove also the biological signal of interest. So that's the design. And here I'll just say a few more words about the different types of methods that we've developed to address this question of stem cell differentiation. So this is a workflow, and I've indicated in gray the different packages we've developed to perform the task. And most of these packages, actually all, I think, are released on Bioconductor. So before we do anything, we look at data. We look at data to make sure we understand our data and we detect the main features, whether they're good or bad. So looking at data is really crucial to avoid egregious problems with the data, to avoid garbage in, garbage out. Sach and Sylvia know that my group spends a lot of time on normalization and pre-processing. This is a bit, the idea here is to basically adjust for unwanted technical effects. And this seemingly mundane step can actually have a huge impact on the results of downstream analysis. Often the impact of normalization can be greater than the impact of the choice of a higher level analysis method. So I'll discuss normalization in this talk. I'll mention dimensionally reduction only here. I won't have time to describe our methods, but as you know, dimensionally reduction is a very common step in high throughput assays to visualize the data, of course, but also as input to downstream methods such as cluster analysis or lineage inference. And principal component analysis is, well-known classical method. It has well-known shortcomings as well, but it's still useful in many of these genomic applications. And in particular, we can significantly improve on classical PCA by taking two types of steps. One of them is to have a regularization step, and that induces sparsity, stability, and interpretability in the principal components. So this is kind of the same idea as you would do in the lasso we can apply to PCA. And when we have controlled data in the form of background data sets, we can also use a contrastive step in principal component analysis to do some normalization of the data. So it's basically performing PCA, but on a modified version of the covariance matrix, which has been adjusted by the covariance matrix of your background data sets. So we've combined these two ideas in this sparse contrastive principal component analysis method, and we have software for that. Also just mentioned this Zimbi wave method. So it stands for zero inflated negative binomial based wanted variation extraction. And in this method, we've basically brought together a range of consideration that we've seen in EDA and normalization. In particular, we use very naturally a zero inflated negative binomial distribution to account for zero inflation and also over dispersion that are well-known features of single-cell RNA-seq data, but also by using a suitable parameterization of the negative binomial mean, we can perform normalization. We can adjust for known and unwanted and unknown factors of unwanted variation. We can quantify the biological effects of interest and also perform some dimensionality reduction. And in particular, the weights that we get from this Zimbi distribution can be used for differential expression analysis purposes. So we can use weights from Zimbi as input to standard differential expression methods like EdgeR, DSeq, or Lemma. So clustering, again, a very common task in genomics. It's used for its own sake for finding novel classes of cells or genes, but it's also very useful for QC and EDA, or as input to other methods, for example, in linear inference, in lineage inference. And then the rest of the methods we've developed are more specific to lineage inference. So in particular, Slingshot to infer lineage and pseudotime, so positions of the cells on the lineages. And I'll discuss that in greater detail later. Also, once we have the trajectory structure, we're interested in finding which genes are involved in the differentiation process. So we've developed TraitSeq for that purpose. And finally, I'll mention a few words, if I don't run out of time, about a new method we've developed. It's called condiments, and this is to look at trajectory inference when you have multiple conditions, like wild type and knockout animals or different time points or treatments. So let's look at data now. So first, just mention the zero inflation issue. So these are bar plots of the per cell proportions of genes that have zero count. So each bar is a cell and the bars are colored by batch. So you can immediately see that over 80% of the genes have zero count in some of the cells. So that's something we need to deal with. Also, for each of our cells, we have quality control measures. These quality control measures reflect properties of the alignment of the reads, but also the sequence composition of the reads. So there's typically, 12 or so that we have per experiment. So here, these are box plots stratified by batch of the QC measures. So the main message here is that quality varies both within and between batch. And I'll come back to that in the context of normalization. More importantly, the quality control measures are associated with what you're interested in measuring. The quality control measures are associated with the read counts. So let's look for now just at the figure on the left. This is a scatter plot of the first principal component of the quality control measures against the first principal component of the read counts. So each dot on the plot corresponds to a cell. And again, we've colored the cells by batch. So you can see here that you have an association between the QC measures and what you're interested in measuring, the expression measures. So that's relevant for normalization. And then the last plot I'll present here in this first EDA stage is the distribution of the gene level read counts. So on the left, we have box plots for each cell, so about 600 cells, of the gene level log counts. So of course, we've seen that in the zero inflation plot that we'll have many zeros. So naturally, the log counts are shifted towards zero. We've had an offset of 1 to avoid taking the log of 0. There's a lot of variation within batch and between batch in the read counts. So that's variation within a batch is technical variation, and it can be masking the biological variation of interest. So we need to address that at the normalization stage. So this brings me to a few general comments about normalization. So the goal of normalization, again, is to adjust read counts for both gene level and sample level unwanted technical effects. So at the gene level, we can have GC content effects. That's, for example, a very big issue when you look at ataxic data. And there are sample level effects like sequencing depth, batch, QC measures. We'll be focusing mostly here on sample level effects. And in order to allow meaningful comparisons of expression measures between genes or samples, we need to perform some type of normalization. So normalization is actually essential before any type of downstream analysis, such as clustering or differential expression, to make sure that when we see a difference in expression measures, this difference is really due to a biological effect of interest and not a technical artifact. And normalization is even more important for single-cell RNA-seq than bulk RNA-seq because of the increased levels of technical noise and the zero inflation. So two questions whenever we think about normalization. So one of them is, does it matter? And here the answer is an emphatic yes. The choice of normalization method can have a large impact on the results, often larger than that of the choice of downstream method. The difficult question is, which method is best? There's no obvious answer here, and it's really data set dependent. And we need data-driven approaches and controls for selecting a suitable normalization procedure. And I feel like there's still much work to be done in this area, particularly in the inclusion of both sample controls and also gene or feature level controls in these high throughput experiments. So we've developed a general framework for normalization that tries to address these issues. So it's called SCONE, and there's the bioconductor package for that. So the first component of the package is the implementation of a range of normalization methods that span that start from very simple global scaling methods like DSeq, TMM, or upper quantile. So a global scaling method is when you divide the read counts by the same number for each cell. So that helps deal with issues like sequencing depth. But the technical effects are typically more complex and require more aggressive normalization. So a very aggressive normalization method is full quantile normalization, where you basically match the quantiles across all your samples. And then there are also regression-based methods where you regress the read counts on known factors of variation, like batch or quality control measures. And then there's also factor analysis model, like the remove unwanted variation method that was developed by Terry Speed and his group in the context of microarrays and bulk RNA-seq. We can also use that in single-cell RNA-seq. So that's implementation of method. And then in order to help choose an appropriate method for a particular data set, we've developed a range of normalization performance metrics that focus on the following three main features of the data: whether the samples cluster according to factors of unwanted variation or wanted variation. So if we get clustering of the samples by biology, this is good. If the samples cluster by batch, this is very bad. Okay. We also look at between sample distributions of expression measures using RLE, relative log expressions. And we look also at the association of our expression measures with factors of wanted and unwanted variation. So we have about 7 measures that we look at that assess the performance of the normalization methods that we apply. And I should say that the SCOAN framework is quite general in the sense that you could apply it to other data. My group has worked on adaptomics and metabolomics data, which have a similar structure, but for which we've used different types of performance metrics. So you can still use abstract, you can still use the general idea, but you have different ways of assessing performance. So we've applied the SCOAN framework to our olfactory epithelium P63 data set. Remember, we had 600 cells and about 600 cells, and we actually evaluated 172 normalization procedures. So this may seem completely crazy, but we have a way of basically selecting among these procedures using our performance metrics. So the procedures we've used range from very simple global scaling methods to regression-based methods that adjust for biology and that adjust for batch. And among the best performing methods were methods that combine full quantile normalization with regression on principal components for the quality control measures, and then adjusting for biology without adjusting for batch. If we had adjusted for batch only, we would have removed the biology of interest because of the nesting. And SCOAN was able to detect that, to tell us that, no, you shouldn't adjust from batch because you'll get rid of the biological signal. So this is one of the main outputs from SCOAN. It's a biplot of the performance scores. So after applying these 172 normalization procedures and the SCOAN metrics, we have data that are in the form of a matrix that have rows for the normalization procedures, 172, and then 7 performance metrics. So we do a biplot of that. So here you have principal components that take you from 7 dimensions down to 2. Each point on the scatter plot corresponds to a normalization method. And we've colored the normalization methods from a blue to yellow scale, where yellow would mean they have a good performance measure. So the good normalization measures are in this corner. So let's see what happened after we picked one of the top SCOAN measures. So these are box plots of the gene level relative log expression. So relative log expression are actually more informative than just log counts. So for a gene, the relative log expression is the log ratio of its read count to the median read count across sample. And the idea here is that when you look at box plots of RLEs, where again here each box corresponds to a cell and the boxes are colored by batch, if the gene level read counts have similar distributions across cells, the RLEs should be centered around 0 and have about the same spread. So here we can see that, as expected, after a normalization, we get much more evenly distributed RLEs than before normalization. These are the RLEs before, and these are the RLEs after normalization for all the genes. And just as a check, we had housekeeping genes in this experiment. So we also looked at the RLEs of housekeeping genes. Those are genes that should be constantly expressed across cells and batch and biological conditions. And they're, reasonably stable. So these are just mostly sanity checks. We've also looked at the correlation of the normalized expression measures with QC metrics. And here we've decreased the correlation as expected because we've regressed out the first 2 principal components of QC measures. So that's all I'll say for now about normalization. Are there any questions, or should I continue with the rest of the presentations? Clarification questions?
I'd say unless there are any sort of really pressing questions, let's take questions at the end. But
good. Thank you, Sach. All right, so I'll say a few words about cluster analysis now. So clustering of genes or sample is a common task in omics data analysis for discovering novel classes of genes or cells. but also for exploratory data analysis and quality control. And also here as an input to other methods, we'll use cell clusters as input to our lineage inference method. But as you know, clustering is a difficult and frustrating problem. No matter what the data or clustering algorithm will get clusters. The question is whether they're real, whether they're replicable, and also how do you validate these clusters. Also, clustering results are sensitive to the data and to the choice of algorithm and associated tuning parameters. And also in practice, when we have these data sets, we try a range of clustering algorithms and look at the data and try to find a way of combining them, of leveraging the information that you get from different methods. Clustering results are also hard to validate. And there's a well-known trade-off between the resolution of the clusters and how replicable they are across data set. Top-level clusters tend to be more stable, but once you start partitioning more and more your clusters, it's less likely that they're going to hold when you get new data from the same system. You get down into the noise. So you may be asking, why another clustering method when there are so many? So it's actually, this is not what we're proposing. We're not proposing a new clustering procedure per se. Rather, given the existing clustering toolbox, we're trying to address two goals. So the first goal, which I'll discuss today, is to provide a general and flexible framework that enables researchers to easily apply and compare a range of different clustering algorithms. And from this battery of clustering results, generate a stable consensus clustering. The second goal, which I won't have time to discuss today, but which my group has addressed in a paper on the Dune method, the software package is called Dune, is to provide a cluster merging procedure that helps you navigate the trade-offs between cluster resolution and replicability across data sets. So let me talk about RSEC now. So this is for goal 1. So this stands for resampling-based sequential ensemble clustering. And our goal here is to generate stable and tight clusters. So this is an ensemble clustering method in the sense that we either aggregate multiple clusterings obtained from different algorithms applied to the same data sets, or we aggregate clusterings obtained from a given algorithm applied to resampled versions of the learning set. And the whole idea here is essentially borrowed from supervised learning. Is like when you aggregate or in ensemble learning, you get more stability. So think bagging, boosting, random forest, and supervised learning. And of course, ensemble unsupervised learning is not new. So we're also building on that literature about ensemble clustering. So a few words about RSEC. So we start from a base clustering algorithm. This could be partitioning around medoid or PAM or k-means, and associated tuning parameters like the number of principal components or the number of clusters k in k-means or PAM, or the choice of distance matrix. We could use Euclidean distance matrix or Manhattan or whatever your favorite distance matrix is. So we start with this base clustering algorithm, and then we produce a single candidate clustering. And then we repeat this process over different base clustering algorithms and tuning parameters. And then from this multitude of candidates, we generate a single consensus clustering based on their co-clustering matrix. And I'll show you an illustration of that in a moment. We also have a pruning step. So again, very much in the spirits of what's been done in the supervised learning literature, in that we merge non-differential clusters by creating a hierarchy of clusters and then starting from the bottom of the tree, working up the tree, testing for differential expression between sister nodes, and then collapsing nodes that have insufficient differential expression. And finally, as is of interest in biology, we're trying to find signatures for the clusters by testing for differential expression between selected subsets of clusters, not all possible subsets of clusters or random pairs of clusters, but we'll use the hierarchy of the cluster of the trees clusters to find suitable comparisons for DE. So here's an example of applying RSEC to our olfactory epithelium data. This is a heat map of a matrix with rows that corresponds to candidate clusterings, and then columns that corresponds to samples. So here we have about 600 cells. And the order of the samples, the cells at the top, is just the order determined by the first clustering. And then we choose the colors using a greedy algorithm in each subsequent row to sort of maximize the overlap. So what we can see here, based on this kind of messy heat map, is that some samples tend to always be clustered together. Like here, this big green band. Likewise, we have a little bit of a pink band, but then other samples are very noisy. They jump around between clusters depending on what clustering algorithm you use. So we use this to build our co-clustering matrix. So a co-clustering matrix is a matrix that's 600 cells by 600 cells in our application, and its entries corresponds to the proportion of times the pair of samples are clustered together in all of our candidate clusterings. So this is a pseudo-color image of the co-clustering matrix. Blue is a very low co-clustering, and then yellow is a high level of co-clustering. And of course, the diagonal should be 1 because the sample always clusters with itself. So you can see right away here from this co-clustering matrix that you have some groupings of cells that jump out. So our next step is to feed this co-clustering matrix to a hierarchical clustering algorithm and build a tree of clusters here. And then we do very much like in the spirit of CART, we have a pruning. We go up the tree, test at each node whether there's differential expression, and then merge if there's no differential expression. So we start with, I don't know, for 15 or so, I don't count very fast, clusters, but then we prune down to 7 clusters by testing for D. And you can use your favorite test of differential expression. I think we used AJAR in this example. So we applied RSEC to our P63 data set and identified 7 clusters that basically correspond to the variety of states we expected in the differentiation process of our HBCs into neurons and sustentacular cells. So we recovered the HBCs, the global spatial cells, the microvillus cells, and various types of neurons, as well as the sustentacular cells. Next, we asked which genes differentiate between these different cell types. So here we have 7 cell types. So what comparisons are relevant? We could potentially think of doing all pairs, right? What genes differentiate HBCs from microvillus cells and so on. We could do a very non-specific F statistic computation saying, are there any differences for that gene between the cell type? That's not very informative, and I'll show you this. These are the results of computing F statistics for all the genes across the cell types. And this is a heat map of the top nearly 500 genes. So genes here, cells here. So no real patterns emerge. So instead, what we did was to use this tree structure and say, let me find genes that are differentially expressed between these 2 cell types, then these 2, then these 2, these 2, these 2, and these 2. And these are the results of these more specific comparisons guided by the structure of the tree clusters. Again, genes here, single cells at the top, And here are the various types of comparison. This is the genes that are differentially expressed at node 1, here at the very top, and then looking at the other nodes. So we get much more interpretable patterns, and this is what we provide our collaborators, and they're able to examine the different types of gene clusters that emerge from this representation. So the rest of the talk will be devoted to lineage inference, finally, addressing differentiation of cell types. So we've developed a framework, Slingshot, to infer cell lineages and pseudotimes. Pseudotimes are referred to the positions of cells along the lineages. So it's mostly in terms of transcriptional activity, not necessarily chronological times, but often transcriptional time and chronological time have a strong relationship. So Slingshot allows us to identify any number of novel lineages. So we don't need to pre-specify the number of lineages, and it can be any number greater than 2 or 1 even. And we can also incorporate subject matter knowledge to supervise part of the inference process. So for instance, here with the olfactory epithelium, we already know the terminal cell types. We know that we expect to have sustentacular cells and neurons, so we can tell Slingshot to use that knowledge to infer the trajectory. So there are 2 main steps here. The first step is to infer the global lineage structure. In other words, the number of lineages and where they branch. And to do this, we use a minimum spanning tree, not on the cells, but actually on the clusters. So it gives us much more stability when we try to examine the global structure of the trajectory. With the cells, it would be too noisy. And also it's much more computationally intensive when you work at the cell level. It's only at the second step of Slingshot that we work with cells. And this is where we infer their pseudotime along each lineage using simultaneous principal curves. So principal curves, in some sense, you can think of them as a version of LOS, of smoothing, where the X and Y variables are interchangeable. Okay. So we applied Slingshot to the olfactory epithelium data set. And we applied it using the cell clusters that we obtained from our 7 cell clusters. And we provided Slingshot with knowledge of the terminal states. So sustentacular cells, microvillus cells, and mature olfactory sensory neurons. It turns out only the first one had an effect, but I'll show you we can get a mistake if we don't supervise the trajectory properly. So Slingshot identified 3 lineages, actually not 2 as we might have initially expected. We thought we might have just the sustentacular lineage and the neuron lineage, but we actually got also a lineage that terminates in the microvillus cells. So in the first lineage, the sustentacular cells are produced via direct conversion of the HBCs, which does not involve cell division. But by contrast, in the second and third lineages, the microvillus cells and the neurons are produced through an intermediate stage, which involve lobose basal cells. And if we had done a completely unsupervised version of Slingshot, we would have drawn the false conclusion, the known false conclusion that sustentacular cells can develop into GBCs. So here's a display of the Slingshot results. The first panel is a 3-dimensional representation of the cells using principal component analysis. The cells are colored by cluster, by cell types. So for example, in green, you have the HBCs. The neurons are orange or red, microvillus cells are blue, and the sustentacular cells are red. So I'm sorry, I got the neurons reversed with the sustentacular cells. Sustentacular cells are red. The middle panel is a representation of the minimal spanning tree, the MST, with nodes that correspond to the cell types. And then finally, the third panel displays the principal curves and the cells that are projected on the principal curves to get their pseudotimes. So once we have the trajectory, a natural question is to ask which genes are associated with the lineages. And this is what gives us some information about the biological process that underlie differentiation. So today, most approaches focused on differential expression between discrete groups of cells, between clusters. And that fails to exploit the continuous resolution that is provided by trajectory inference, the pseudotimes, right? So what my postdoc, Koen van den Berg, we introduced TraitSeq, which is a generalized additive model framework based on the negative binomial distribution, which allows us to look at both within and between lineage differential expression. And also using the fitted values from the negative binomial GAM, we can cluster genes according to the expression pattern. So the idea here is that we fit the GAM and then we identify different types of differential expression by looking at contrast between the GAM coefficients. So here's a quick description of the negative binomial GAM. G corresponds to genes, I to cell, Y is an expression measure, T is a pseudotime, so it's specific to a cell and a lineage. And we parameterize the mean of the negative binomial GAM using cubic splines for the pseudotimes, covariates, cell level covariates like batch, and then offsets that could correspond to sequencing depth. And the parameters of interest in that framework are the beta, the coefficients in the splines. And depending on what contrast of the betas, the GAM coefficients we look at, we can test for different types of differential expression patterns. So just a cartoon here. This is one gene, okay? Is the scatter plot of the log count for that gene against the pseudotime for that gene. This is a simple example where we have two lineages, an orange lineage and a blue lineage. For that gene, there's differential expression along the blue lineage, but not along the orange lineage. So depending on the contrast that we look at within NBGAM, we should be able to detect differential expression within lineage for the blue lineage, but not for the orange lineage. For testing for differential expression between lineages, we should be able to tell that there's a difference between the blue and the orange lineage. So in the current implementation of TraitSeq, we provide two types of differential expression within lineage, and then three types of differential expression patterns between lineages. So again, it's all based on looking at suitable contrast of the betas. So I'll end with just the presentation of the results of TraitSeq on our olfactory epithelium data. So again, this is what we've seen earlier. It's the three-dimensional representation of the cells, and the Slingshot lineages are HBC to sustentacular lineage, HBC to microvillus lineage, HBC to neuronal lineage. These are just the different types of cells. And here in panel C, in the top, in the bottom left pattern, we have a heat map of the top 200 genes that were found to be associated with the neuronal lineage, with differential expression in the neuronal lineage. And we can see that there are 5 groups of genes that emerge with distinct waves of expression, early expression in that group, and then middle, and then towards the end of the lineage, high expression. So this is a within lineage D pattern. In this final panel, we looked at between lineage differential expression. So differential expression using the early DIA test. So we asked, can we find genes that are differentially expressed early on between the 3 different lineages? And this yielded 4 transcription factors that seem to be differentially expressed early on between the 3 different lineages. So SOX1, ELF, BLC, 11B, and then BB. So it turns out that all 4 of these transcription factors are involved in epithelial cell differentiation. So you know, I won't have time to elaborate on the biology, and my collaborators would do this better than I do, but by being able to provide them with lists of genes that pick up different expression patterns, they were able to understand better how the cell sums differentiate. So I know we're running out of time, so I'll stop here, and I'll thank you very much for your attention, and I'd be happy to answer questions.
Thanks so much, Sandrine. It was really, really great. And yeah, we have a few minutes for questions, and so please feel free to just put your hand up or put the question into the chat, and we can get. Let me kick off with one question straight away, Sandrine, which is you know, I like the emphasis you put on data normalization. You know, the fact that, you know, there can very typically, you know, the normalization is going to have more of an effect than you know, which downstream analysis method you use, and then that's sometimes neglected. But do you have a view on? So there's a view that's sometimes expressed that for supervised learning, particularly with sort of, you know, modern, very sort of flexible methods, you somehow don't need a normalization step because the methods are so flexible they can sort of figure out the appropriate normalization against the target label. In any case, do you have a view on that? I mean, do you think normalization methods should always be used, even in the case of supervised learning, or is it sort of okay to let the supervised learner figure it out?
I have a feeling we still need them, because if you think, I mean, if I take a specific example of supervised learning, if I were to build a classifier for the cell based on the differentials on the gene expression, I would still have data. The data that I would feed to my classifier would have still some technical differences between the cell type, and then may mislead my classifier. I think a question that's still very much open for debate, though, is how much of the normalization do you do upstream, as opposed to how much of the normalization do you do as part of the particular method that you apply. So you can think of using classifiers that explicitly account for the batch variables, say. And in that case, that may be sufficient, right? So a very simple classifier, you could have your class label, but add also a factor that corresponds to technical variables that you know are present, technical effects that you know are present. And that's, I don't really have a good answer to that. I think the answer might be more like something in the middle. Yeah, you do need some upfront normalization at the beginning, but you could probably improve on that by using a method that's really specific to the question you're addressing. So if it's classification, you might be able to really get some mileage out of building a normalization component inside of your supervised learning method. I could see that being quite fruitful. But
thanks. Thank you very much. We have a question from John Whitaker. Over to you, John.
Oh yeah, thanks for the talk. It's really interesting. So a couple of things. One is the extent to which the sources of variation are known or identifiable, and therefore might be dealt with in other ways, and in particular might be designed. The experiment might be designed in such a way that you minimize them, and maybe minimize the need for so much normalization. And the second is a bit of a cheap question, which is about model fit and how well the negative binomial assumption works for these data. Because I have seen some single cell data which it isn't great, actually.
Yeah, thank you very much, John. So those are two really really, really important questions. The first one, I would love to see more care in experiments in terms of having components that could reduce the need for normalization, or at least help with the normalization. So things like, well, obvious things are experimental design, right? This is an old field that's underused, I think, in these settings, right? But also using knowledge specific to the technology and the biology, like have more control genes, have control cells, control samples. I think that could help us greatly, and then avoid having to take these not ideal measures that we do take. So about 10, maybe more than that, there were these spiking controls from the External RNA Control Consortium. It was dilution series. I thought it was perfect. I mean, the concept to me seemed perfect, but unfortunately in practice, the controls were very noisy, and using them introduced more problems than not using them in some cases. But I would love to see more work being done in that area. Model fit, right? That was your second question. It's true. The negative binomial is not suitable for all data sets that we've had. So I really didn't address that today, but it's really important to look at model fit before you settle on one of these distributions. So it's not a one size fits all.
Okay, and if you're not going to use that, I mean, do you have suggestions of where we might go next? Because we didn't get beyond that. We sort of
Well, I'll probably butcher the quote, but all models are wrong, but some are useful. So I think we're in my experience working for my collaborators, so we're still very much in a hypothesis generation phase, right? So I think being able to produce intermediate results that will later be confirmed is already useful in itself. Having the perfect model, the perfect error control. I mean, I think I've mentioned we're doing testing, but for somebody that wrote a book on multiple, I'm not using any of that anymore. I mean, I look at p-values more at summary statistics that help me produce a ranking that my collaborators can use. So my message would be just keep looking at data. See whether it makes sense. We don't do enough of that. As mentioned, I feel we try to jump right away to modeling. We're comfortable with math, but I think at each stage, look at data, talk with your colleagues, your collaborators. And
I think we probably have time. This is, for maybe
Sylvia, I see us.
Three more questions. So maybe Sylvia, then Scott from the chat, and
I'll have
Mohammed
to look at the
Shafiqur
chat.
Rahman.
Sorry, I didn't look at the chat.
So Sylvia,
Sylvia.
Very quickly, what I was interested in your clustering and your sequential resampling clustering. I was wondering, so you're doing that within one data set, basically. And I was just wondering whether doing that and getting this stable cluster, have you ever looked whether it makes them more transferable to another problem? I mean, do you feel this is a step towards that, or this is just still conditional on this particularly, you're after a stable, reproducible thing for this particular data set?
I'm delighted you asked that question, because that's what we tried to address with this other methods, which I didn't have time to present. The question was exactly what you posed is, it's fine. We have a data set, the clusters are stable, but what does that mean? Can we use them in another context? So we tried to address that with Dune by looking at how stable, how the clusters work on another related data set, and that's really the test, and that's
okay.
It was a preliminary step in that direction, but otherwise we keep using the same data set, and
I will look that up. Thanks a lot, because I think this is something we've been really pondering for some time. This is a hard problem, but it's great that
it's a small step in a large problem. There's a lot more to be done. So thank you for asking that question.
Okay, Scott from chat, then I think Mohammed Shafiqur, then Camilla, and then we'll probably have to close after that. But Scott, would you like to ask your question directly, or should I read it out?
That's a very good point. So question one from Scott: do the ensemble clustering methods provide some sort of confidence of clustering metric? So we looked at that. So I think, yes, they can, definitely, in the sense that you could look at proportion of times samples are clustered in the same cluster. So they're, again, very much, metrics are very much inspired from what we do in supervised learning, where you have votes, right, when you do classification. So we don't have that in that paper, but an earlier paper that I wrote back in the microarray days with my collaborator Jane Friedland addressed that. We can get by looking at how frequently the samples stay in the same cluster. So that's a very, very good point. Two, do they somehow allow for diversity of clustering algorithm? A consensus between two very similar algorithms would be less convincing. That's then a consensus between two quite different algorithms. That's an excellent point, and we did not address that, but I think, I'm just thinking out loud here, I think the framework could allow you to be more specific when you combine them as to, oh, here I'm combining a bunch of clustering algorithms that are very similar, but compared to comparing clustering algorithms that are very different. So you could potentially think of having weights for how you combine the results that exploit that information. So that would be something very interesting to consider later on. Thanks for bringing that up, Scott.
Mohammed Shafiqur Rahman, if you'd like to ask a question.
Oh, I can't hear very well.
We can't hear you, unfortunately.
Can you hear me?
Yes.
Oh, yes. Now, yes. Thank you.
I find that trajectory analysis very interesting. I was just wondering, like, how robust is the analysis, and if it's possible to do trajectory analysis, then we might not need any controls, do we? Or can we only use the cases and see how gene expressed through the trajectory?
I'm not sure. So the controls, you mean control samples or control genes?
Yeah.
What would be the controls
control
here?
people or cases and controls, and we can use only cases, can we?
I think it depends on the question you're trying to ask. You could do the trajectory analysis on if you just want to see how stem cells or cells differentiate in controls, you could do it in controls. If you care about studying how stem cells differentiate in cases, you do in cases. And then maybe the next, this is what I didn't have time to present, is you could ask yourself how this was condiments. The next method I was about to present is you may be interested in seeing whether there are differences in the trajectories of differentiation between cases and controls. So you could do a combined analysis of the two. I think that's, yeah. The stability is a very good one, and we haven't done that much on that end yet, like stability to cells. So you can think of subsampling cells and then seeing how stable the structure is. So that's something we haven't really done. So I'd be curious to see more of that. Thank you, Mohammed.
Thank you.
Maybe a last question then from Camilla.
Oh, hi, Camilla.
Yeah.
Nice to see you.
Yeah, can't believe it's been probably 4 years now.
It's lovely to know that you're over in Cambridge.
You opened my eyes for bioconductors, so it's fun to see this. I've used it a lot since back then, and it's such a neat workflow. I really appreciate it. So it's not as much of a question as kind of a comment that I work a lot of network models, and one thing that I've always been missing is just a really neat way to perform kind of validation of networks. So either enrichment analysis or just checking up the databases. And bioconductor is great for, accessing lots of data sets. So that's just something that I would love to see in the future.
Oh, that's good. Thank you for bringing that up. It's lovely. So Camilla was a student at Berkeley.
Yeah.
A few years ago. So I was like, oh,
I know.
Wonderful to see you.
I was happy to see you were having this talk. It was really fun to see you again.
Yeah. Nice surprise to catch up.
Okay, fantastic. Thanks everyone. Really, really interesting discussion. from which I take away that there's still actually open questions at this intersection between sort of unsupervised exploratory learning, sort of validation thereof, normalization, etc. That it's not completely settled as yet. But thanks very much, Sandrine, for really stimulating talk and really taking us through the sort of workflow and not just sort of, you know, one method and you know, the usual sort of things. I really appreciate that. It fits very well with what I think we're trying to sort of achieve with the lecture series. So thank you very much for your contribution. It's been a pleasure.
Well,
thanks everyone.
Thanks everyone. I really appreciated the question, and then it was great to catch up with people that I knew from before. Lovely. Thank you so much for attending.
Thanks very much.
Take care. Bye-bye.
Take care. Bye.